So today I'm going to talking about the subseafloor biosphere. So my field is subseafloor. So then what we use is this kind of the vessel and lowering the pipe and drill down to the seafloor and to get a sample. And we sometimes see this kind of the tiny, cute guys. And so for obtaining a sample or bringing a ship to the center of the ocean, the international collaboration was pretty much important. And that is why we have the IODP, so to say, the International Ocean Discovery Program. I cannot have a better control of the mouse, but it is set IODP. And having the United States and Japan and the European countries collaborating together to get us samples and gathering or collaborating our knowledge to find every hidden mystery beneath the seafloor. By the way, can someone say that how deep have human drilled down from the surface ever? Can you? Yes, please. Great. Yes, 12 kilometers at the Russia. And that is the deepest record we have human ever drilled down. And thinking about the altitude the space station locates, it is 400 kilometers. And thinking about half a diameter of the Earth, so let's say 6,000 kilometers. So then you should know that we only scratch the surface of the Earth. Even beneath our feet has many of the unknowns. So then such an international collaboration was important. And we uses the various vessels. And maybe some of you heard about this sad news that the US drilling ship, one of the flagship drill ship of the Joyless Resolution, recently retired. And, but we still have the Japanese vessel, Chikyu, in the center. And we are about to begin the new international ocean drilling program. We are about to start to this new project, which is the continuation of the international collaboration in the future. So then, going to the deep biosphere. So back in 60 years, 70 years, our knowledge about the deep biosphere was only the 7 meters below the seafloor. So someone put the metal pipe onto the sea surface. Not the sea surface, but the sea floor, and took the sample and do the plating and cultivate microbes. And what they succeeded is in the down to 7 meters below the seafloor. And at that time, 60, 70 years ago, the conclusion was the end of the biosphere sits in the 7 meters below the seafloor. But now we know that more than 2.4 kilometers below the seafloor, still microbes are there. And in addition, they are living in such an environment. And thinking about the global biomass, subseafloor sediment harbors the roughly 1/3 of the entire Earth biomass, or microbial biomass, so to say. So this comprises the most abundant microbial biosphere in the Earth. And also their diversity is as large as the soil and ocean microbiome. So and also they are distinct in their community. I'm still having a problem, but yeah. So here's the marine sediment microbiome, and you can look at as the very different color from the others. So then, yes, thank you. Okay, so then, and if you do the some special plot, you see the subseafloor sediment and the marine and soil has the distinct communities, each other. And also, yes, the diversity says that the marine sediment is having the equivalent diversity to the ocean and the soil. And I joined JAMSTEC 18 years ago. And at that time, my recognition of the subseafloor biosphere is something like this. But soon I found this is completely wrong. Why is that? Is the major matrix of the subseafloor sediment is the non-biological grains. And the microbes are only the minor component. That comprises only the less than 0.01%. And the other problem is that these guys, even containing amino acids, proteins, or DNAs. So even if you find amino acids, proteins, or DNAs, this doesn't mean in the subseafloor world that you find life, or it can be the case that you find only the past life, already dead or something. So we have to have the way to access microbial cells. So then my career in JAMSTEC was the continuation of the technical developments to drive discovery. And today in this symposium, I want to share my experience of the failure for the younger generation people. I still have kind of a trauma that, that I made potentially goes to the very critical failure in my early career. But I could solve such kind of a problem. So I want to tell about that story. So this is a picture I took 18 years ago. And with that, I did the DNA staining by using the reagent called as a cybergreen one, which is known as the DNA-specific compound. If you stain your sample with the cybergreen one, you can easily get this kind of the picture. Green dots is usually considered as the DNA-containing microbes. And which ones are microbes? Can someone tell? I asked supervisor at that time which one I should count as a microbes, and then he said, it is DNA stain, and you see fluorescence, and that should contain DNA. So then count them all. And result was something like this. If you have seen any kind of the subseafloor microbial distribution, it is very strange. But it was 18 years ago. No one knows whether it is strange or not. But looking at the membrane, I prepared a membrane by filtering the sediment sample onto the membrane and the staining I made. And on the same batch, different portion of the membrane, I tried to look at on the electron microscopy, scanning electron microscopy. And the result is something like that. Where is microbes? I thought microbes could be visualized something like this, but I didn't find anything look like that kind of microbes, which made me so nervous. But with that data, my supervisors were so excited. It could be a groundbreaking result, and you should continue to pursue what is their composition, or something like that. But even looking at this fluorescence microscopy, you see this type of the triangular shape, not look like a microbial particles still remain. And having the SEM image, I got nervous and nervous and nervous. But we created this type of the figures by the many of the experiments I conducted. So let's say not only the cell concentration, we measured the carbon content and the isotopic content, and even the nitrogen content we measured. And it is actually it was submitted to the science. Fortunately, it was rejected, but it was kind of a nightmare. Now you know why I have trauma. So then, the other stuff I had, this guy. So I took this type of the picture by the SEM observation. And one day, when I look at the TV program, JAMSTEC president was there, holding this picture. And he said, "This is subseafloor archaea." I didn't say anything about this is even the microbes or not. I didn't say anything, but he holds that picture in TV show, says that the subseafloor archaea. Wow. So it was terrible experience. I screamed even. So then, I was nervous and even terrified by the result I had so far. And then I thought how to solve this problem, and by coincidence I found this. So that the matter was the concentration of the stain applied. So if we apply cyber green 1 reagent to the higher concentration, then suddenly I saw some kind of the difference of the fluorescence color, like this. So in the arrow, you can see. Then now we know that the DNA cyber green complex has kind of the social distance each other, each molecules, and then stay green, although in a case of the over-absorption of the cyber green 1 reagent onto the non-microbiological grains, they are close each other, and the kind of the energy transfer can happen. So that is why, so this type of the spectral difference we found. So it was kind of the coincidence, and we could utilize this type of the findings towards discriminating the microbial cells and sediment particles, non-biological particles, and even could establish kind of the image analysis to look for only the greeny material, greeny particles, and excluding the yellowish fluorescence particles, and then got this type of the figures relieved. So this is type of the the figure we usually see in the subseafloor sedimentary microbiological abundance. So then, what did I learn from this failure was it was critically important that we need to understand subject and appropriateness of the methods, and the other stuff is supervisors can be wrong. And we can be at conference even without kind of the validation. What about paper? Some kind of the the mispublication can happen. That's why we have the system of the retraction. But if someone do not do retraction, what happens? So it will remain in the history forever. So that is a problem. And it was, I said, just a coincidence to find a problem, but it was very important for me to keep questioning by myself, something is wrong, and what is wrong or something. And by having such kind of the trauma experience, I got somehow careful to do something, but we also explored our methods by incorporating new things. One is this type of the density-based separation of the sediment particles and microbes, and employing, so by using that before separation or concentration, subseafloor sediment looked like this, but after separation or concentration, we can have the concentration of the microbes. And also, we are concerned about contamination as well. And in the geomicrobiology field or subseafloor microbiology field, I'm known as a person who is crazily concerned about contamination, but I'm happy to hear this morning session. I find someone who is more crazy than me. I'm happy. Thank you. So then, what I concern is the excluding the contamination from the terrestrial environment. So this particle counter is not good in quality, but we have the fancier version of that. And actually, inside of this laminar flow clean room, it can create something like a wall of the air, and it can exclude the contamination from the wall. So then, as far as we are facing to the wall, we can have cleanest environment for doing our experiment. And also, oh yeah, so here I placed single microbe on the screen. Yes, at the center. So with the clean environmental condition, we can have the very good quality of the experiment. Like the on average, in the blank control, we only have the 4 plus minus 4 cells per cubic centimeter. So which gives us the 16 cells per cubic centimeter of the sediment as a minimum quantification limit, which is kind of the world record, I think. No one have ever beat us. And then, so for doing that, kind of the quality assurance and quality control was necessary and important. And also we are concerned about static electricity. So if you have static electricity, you have attraction of the particles onto your surface. So in the environment, we are having onshore lab and on board, we have the ionizer or neutralizer here and there to neutralize the static electricity to avoid contamination problem. And with that, we could have the good technology or enough technology to look into the deep in the subseafloor environment. With that, we could explore pretty much oligotrophic environment and very deep down to the 2.4, 2.5 kilometers down to the down from the seafloor, and even high temperature up to 120 degrees C. And not only finding, but we want to know what is their living status of the microbes. So then, since we can have some kind of sample in the certain mass, so then we could do kind of the incubation experiment. So this is kind of the experiment we have done. So having the samples in a glass vial and put the stable isotope labeled substrate, put on that sediment and incubate it. And after that, we look at the microbes, and also not only concentrating microbes, we fluorescently activated sorted to further purify microbial cells, because the sedimentary particles can contain organic matters, and even that is non-biological grain. So that is why the kind of the fluorescence activated sorting, looking at the greeny fluorescence color stained by cybergreen 1 was necessary. And having it, we could do nanoscale secondary ion mass spectrometry experiment to capture the isotopic distribution in the nanometer scale, which is good enough for looking at the single microbial cells. And then let's say having the 12 carbon and 13 carbon and 14 nitrogen and 15 nitrogen data, we can have this type of the data showing the single cell level incorporation of the added substrate. So then what we found is that the microbes in the up to 100 million years old sediment were alive and ready to eat their food. So this shows upper part shows the bicarbonate and ammonia added condition, and the lower part is the amino acids added condition. So it clearly shows that the subseafloor microbes are loving the amino acids. So they are eager to eat. But still, there is some cells eating or assimilating carbon dioxide, which was interesting. But it put the further proof that the previous discussion that the subseafloor microbes are mostly comprised by the heterotrophic microbes. And we know that more than 70% of the subseafloor microbes remain alive, sometimes more than 90%, or majority of them are alive, and they are readily metabolized added substrate, we found. And it is published in 2020, but it took 1.5 years to get through the review process. Once rejected and we rebutted. But one of the critical points the reviewer said that they were concerned about contamination, of course. And then we proved that this type of the amplicon sequence analysis, so which kind of the microbes are present in our incubated sample and the prior to the incubation sample, and we could show that this circular one was the pre-incubation sample. And after incubation, the microbial community didn't change so much. So then it was one of the proof. And the other critics that reviewer raised was have they been there since burial? So even though you look at the 100 million years old sediment, it could be the microbes who recently joined to the such a sedimentary biosphere. To prove that, we found this thick porcelainate. Porcelainate is some kind of the ceramics, and which has a very low porosity. So that sits in somewhere here, and our 100 million years old sediment is located at here. So this is almost impossible that the younger age of the sedimentary microbes can migrate through these thick layers. And the calculation of the average pore size says that in median, the 0.02 microns, so which is 1/10 smaller than usual sterilization filter. So it says that the cells sitting in the sediment could have been trapped entire history of that sediment experience. So the 100 million years almost. So like this. And then, so I showed this cartoon. So somehow healthy, look like, got starved and very exhausted. So something happening like this in the subseafloor habitat. So even at the ultra-oligotrophic environment of the subseafloor, microbes are alive. But how it's possible is still a question. By looking at the maintenance power found in literature, says that this type of the energy, so lowest at what? So 10 to the minus 18 watts. But in the case of the subseafloor environment we investigated, the energy availability was calculated to be a zeptowatt level. So 10 to the minus 21. So far below the any known maintenance power. So even in the bioenergetics paper of the subseafloor environment, authors need to include some of the natural mystery to enable for the microbes to survive in such an energy-starved condition. And in yesterday's discussion, someone said that the life as a definition requires the Darwinian evolution. But what about these guys? Left at the very energy-starved condition, unable to replicate, even the maintenance power missing, are they able to evolve Darwinian? It is still a question whether we should say this guy as life, but they could revive after getting to the surface. So it should be regarded as life, but the state in the subsea floor is still a question. One of the other interesting papers appeared recently says that radiolysis of the water could provide hydrogen to the subsea floor microbes. So this could somehow complement the lacking energy for the microbes. And I don't have quite good enough time to say or explain about detail of these papers, but we also investigated in a very hot subsea floor environment, and we did something like the transcriptomics and the metagenomic assembled genome study, showing that the some kind of the active member of the microbes are living in the some depths of the subsea floor environment, and also some kind of adaptation is occurring at the certain depths of the subsea floor environment. And so then I want to summarize that what we can do, or what is still challenging in the subsea floor biosphere study. So in a case, we have a sample with the 10 to the 6 or 10 to the 7 cells per cubic centimeter. We can do many things, but decreasing population bring us to do less things. In a case of this type of the sample, even though we can do kind of the sorting and the concentration of the microbes in the 1,000 or 10,000 number, we still have problem in the amplifying DNA out of that sample. So kind of the density separation or sorting to purify microbial cells together with the enzymatic restoration potentially effective, and this is kind of the ongoing work. And also something escaping from our staining appeared. So detection of the life-specific signatures by us are mainly done by the staining of the biomolecules, DNA. But staining has an intrinsic problem of the non-specific binding, which was solved, but the dye penetration was another problem. So in the case of this type of the sample was okay, but in the case of even the cultivated spores of the microbes are resistant to the stain. So they stay in the yellowish color, which shows we cannot discriminate whether they are living life or just a grain. Then we find out some of the potentially useful techniques like the oramine, which is a compound to stain microbes, spores surface, which is useful to stain the pure culture of the spores. And also we are employing the multiplex cards, so Raman, non-linear Raman, to find the signature of the cultivated guys. And also we had a situation that something like a discrepancy of the stainable life and the spores. So they are obtained by the staining and the chemical detection of the living signature. And we found that this kind of the discrepancy zone. But applying the oramine could stain some kind of the microbial cells, which should be spores, but this is still the ongoing study. And this region we found, so this type of the analysis could be one of the clue to find another microbial dark matter in future. So then the things we have learned and remaining challenges is something like this. But so we find that the old 100 million years sediment microbes, but we haven't identified the end of the biosphere yet. And we also have the question how the microbial community give the global significance to the ecosystems of the Earth environment is still unknown. But I'd like to concentrate on this end of biosphere discussion from now on. So in the past, we investigated or we tried to reach the end of the biosphere through our subseafloor drilling mission. So several attempts have been made, but we haven't reached yet. So in a case of this very deep subseafloor environment, even though we could reach down to close to 2.5 kilometers below the seafloor, but the temperature was still 60 degrees C. So, and it was too deep to drill further, so we couldn't drill further. And in the case of the other expedition, we drilled the offshore Shikoku Island. In this case, we reached to the basement, so basement rock, so which is not appropriate to look at the end of the biosphere. But even though we reached the 120 degrees C, we still find the microbial cells. And in the case of the Guaymas Basin, the basin we find at the California Californian Peninsula. And so at the time, although we reached also towards close to the 120 degrees C, we encountered the hydrocarbon alert, so which was dangerous. We cannot drill further. So someone blocks us to get higher temperature. And in the case of the extremely high temperature region at the hydrothermal field of the Okinawa Trough, even though we reached to the very high temperature zone, that time variety of the temperature zone was so large, so we cannot say what is the clear boundary of the biosphere. So why we are looking at the microbial or the biosphere fringe in the subseafloor environment is like this. So after formation of the oceanic crust, the continuous sedimentation creates the subseafloor sedimentary environment. And throughout the history of the sedimentation, the temperature, chemistry, and pressure changes along the time. And also the oxidation reduction potential also changes. So this is kind of the very good natural experimental chamber for the evolution and the challenge of the life at its fringe condition. And then we are aiming to find somewhere we can reach at the 160 degrees C. For sure, biosphere ends somewhere interior Earth. Of course, interior Earth should be 5,000 degrees C or something, so which is far beyond the life existence. And highest cultivation temperature of the microbes is 122 degrees C, done by our colleague Ken Takai. And the stability zone of the protein is known to be up around 148.5 degrees C. So having the safety margin of the temperature gradient estimate of the heat flow, we thought aiming to 160 degrees C would be good. Yes. I'm about wrapping up. Thank you. So then environmental constraints. The environment needs to be a sediment because sediment can block the microbial migration and they can stay there, and the environment changed. So this is kind of the very basic setup of the experimental chamber. So such kind of the environmental setting is necessary. And no high organics is important to not to reach the hydrocarbon alert. And high heat flow means that the steeper gradient of the geothermal gradient is important to reach somehow accessible region. And easy EEZ is another importance as a regulation. So in a case of the difficult country, we cannot bring any sample to Japan or any other countries. And what we can expect is the observation of the biosphere termination and its factors governing limits. And also the microbial communities shift to the limits, and transition to a lifeless condition mode of the diminishment in fringe will be observed, we are expecting. So then we are listing up variety of the potential zone with depths, and we are trying to locate suitable locations and aiming to submit the workshop proposal or pre-proposal to IODP Cubed by the first deadline. And we are pretty much happy to have your input and for not quite site selection for you guys, but expanding science and collaboration ideas will be pretty much welcome. And this is the last announcement that the JDESC is the Japanese Reading Scientist Consortium, is now giving the course of the program of the education. It is called the JDESC Core School. And last year, we hold the first time of the deep life course, which shows or teaches how you can stain microbes in the difficult sample, how you can get the concentrated microbes out of your sample, something like that. And one of the audience joined, but I cannot find Hirai-kun, but he joined and he enjoyed. And this is it. Thank you for your attention.